Well, welcome to me. Thank you for having me to talk about platinum-resistant ovarian cancer today, which is sort of near and dear to my heart. As really primarily as a drug developer and early phase clinical trialist. This is the space where I work quite a bit. These are my disclosures. Okay, so I kind of broke this up into 3 segments. First, I'm just going to give an overview of the current state of affairs for treatment options in tumors that are no longer appropriate for treatment with platinum. And we term that platinum resistant or inappropriate for platinum. Then I'm going to talk about antibody drug conjugates, and I'm going to end with a little bit on immunotherapy, and I'm going to try and do that in 20 minutes. So when we talk about tumors that are resistant to platinum, that is actually a very broad term, but in general, the pure definition is that these are tumors that grew while you were receiving your last platinum therapy. But it also includes tumors that maybe responded, maybe they shrank to the last platinum therapy and then grew back relatively quickly. And we've lumped them together for a long time, those 2 groups, but they actually are quite different. And I'll talk about why that's becoming more important to differentiate as I talk about some of the clinical trials. But irrespective of how you group or don't group, once a tumor becomes resistant to platinum, it does become more difficult to treat. Not impossible, and we have increasing options, but it does become more difficult to treat. And the figure on the left-hand side is old data, and I think we should update this in a few years with some of the newer therapies, but it is what we have. It does represent relatively recent history that with each regimen that a patient receives for tumors that are resistant to platinum, the expectation for that tumor shrinking goes way down. And the time of disease control on that regimen, even if it doesn't shrink, does it keep it from growing, gets shorter with each line of therapy. And so our patients are starting each line of therapy with more disease present, which causes symptoms, and with more kind of collateral toxicity from whatever we have given them in the past. And so we really are always trying to balance what we're picking next relative sort of to the toxicities a person brings in, so that we're not compounding neuropathy on neuropathy or low platelets on low platelets, for example, but also trying to balance the medicines that we think are going to work best. So this is an area, you'll hear me say this a lot, of high unmet need. These studies in this table are very recent studies, with the exception of the bottom one, which I have a separate slide to show you in a moment. It's sort of the benchmark study in platinum-resistant disease called Aurelia, and it was done over a decade ago. Everything above that, the top 4, are recent clinical trials. And what I'm showing you are the control arms of these clinical trials. So it would be a new drug or combination versus standard of care, which is single agent chemotherapy once a tumor has been exposed to bevacizumab or Avastin. And so what you can see in the column called ORR, that's overall response rate. I'm going to show you what response rate means in clinical trials in a few slides, but I think you can just see whatever it means. These drugs are not very good. They do not get tumors to shrink to the degree that we want them to shrink. And they don't control the disease for as long as we would want them to. It's a little less than 4 months on the average. And so this is sort of what we have in our toolbox. And some patients do quite well with these, and some patients don't benefit at all from these, which is, again, a reason why this disease state is very high unmet need for new and active therapies. Now, this is what's called the NCCN guidelines, National Comprehensive Cancer Network guidelines. Dr. Fader is actually a member of this guidelines committee, and it lists the medicines or combinations that are either FDA approved in ovarian cancer or whatever cancer, or have enough evidence that they can be listed, and it's likely that insurance will provide them. So it's a kind of a mix of things. But it's the list physicians go to when we don't have clinical trials open to see what we can use. So it is a very important list. And you can see the preferred regimens are there in green. And of course, we always want to look at clinical trials. A lot of chemos, that's what cytotoxic chemotherapy means, chemotherapy. And at the bottom, you see 2 targeted therapies, although one of them is targeted chemotherapy, that's mirvetuximab, now known as Elahere, and the other one is bevacizumab, and you know that as Avastin. And so we'll talk through these. The Aurelia trial set the standard of care for early development of tumors that are resistant to platinum. So this clinical trial was done over a decade ago for women or patients with ovarian cancer that was resistant to platinum in the first 1 or 2 lines of therapy. So not 4th and 5th line, which is a lot of patients I see. Early development of platinum resistance, and the patients were randomized to 2 arms. One arm was those chemotherapies I just showed you, pegylated liposomal doxorubicin, we call that Doxil, weekly paclitaxel, or topotecan, versus any of those 3 drugs with bevacizumab or with Avastin. And what you can see from the progression-free survival curve in the middle, in the green, that's the group that got bevacizumab, and in the gray is the group that just got chemo. So I think you don't need to be a statistician to say those look different, and the patients who got bevacizumab did better. And on the bottom in the middle, you can see the overall response rates. And so for patients who got weekly paclitaxel plus Avastin, their response rate was 50%. So that is really, I think everyone agree, our leading option if we can reuse weekly paclitaxel, which sometimes we can't because of neuropathy and travel, because it's weekly. But if we can, that's the leading regimen. And it works pretty well for a period of time. And then it doesn't, and we need other things. But this is the standard of care. The new option for standard of care is mirvetuximab. This is an antibody drug conjugate, so it's targeted chemotherapy. So on the figure on the left-hand side, you can see a cancer cell. Cancer cells are covered in proteins, all sorts of proteins, and they do different things. Some of these proteins, when you bind something to it, the protein basically gives whatever is bound to it a hug and sucks it inside. It's called endocytosis, but it gives it a hug like an amoeba and brings it in. And that's what happens with antibody drug conjugates. You build an antibody that targets a protein that is on the tumor and not on normal cells, ideally, because you don't want to deliver chemo to the normal cell. And you target it to the cancer cell. And on the end of the antibody drug conjugate, and you can see, I don't know if you can see my cursor. Should have brought up a cursor. Anyway, hopefully you can see my cursor. These little red dots are the chemo molecules, not the chemo I just told you about. It's not Doxil or paclitaxel. This is called DM4. They're highly, highly, highly potent molecules of chemotherapy that you can't give as free drug. So when this antibody drug conjugate is rotating or circulating through your bloodstream, it's inactive. Doesn't activate until it binds. It gets sucked into the cancer cell. The cancer cell then dissolves it and releases the chemo payload, and that kills the cancer cell, ideally. Then you can see down here, some of the chemo gets out and gets to the other cancer cells, even if they don't have, in this case, a folate receptor. That's the protein, and it kills them too. That's called bystander effect. That's how antibody drug conjugates work. There's a bunch of these under clinical trials. In ovarian cancer and a whole bunch of solid tumors, you're going to hear a lot about these, but this is mirvetuximab. It targets folate receptor alpha, and it has a chemo on it that's called DM4. It targets microtubules. It was FDA approved based on the SORAYA study, which I've listed for you here. This was a single-arm study of 100 patients, and it was an overall response rate endpoint in a high unmet need. So all of the patients to come on this study had had prior bevacizumab. And so their options would be all those single agent chemos that I showed you that don't work very well. So because it's a high unmet need, the FDA lets us go for something called accelerated approval, which lets us get a response rate endpoint first. And you have to show it lasts a little bit, can respond and then grow. And then it makes the drug available in the US quicker. So patients who have high unmet need can access it. So that's what was done here. The SORAYA study, in the red boxes, you can see the response rate in patients whose tumors had high expression of folate receptor alpha. That's about 40% of serous tumors. So it's not all. Have high. Almost everyone has some. but high 40%. And you can see the response rate is about 32%. So that's a lot better than 13%. And it lasted on average for about 7 months. So that's a lot different. So because that looks a lot different than what available medicines are, it got accelerated approval in November of last year and became available. And so testing for folate receptor alpha on tumors has become a standard part of panels now, like the KERAS panel and other panels. But for tumors that were tested before November, we have to send those separately. So we know the folate receptor status of our patients' tumors, so we can make sure they have access to this medicine if it's appropriate. So this became accelerated approval. Accelerated approvals have to be confirmed with big, long phase 3 studies though. That was the Mirasol study, which I got to lead, so I'm proud of it. This was a big phase 3 study, only for tumors with folate receptor alpha high. They didn't have to have bevacizumab though, they could. And it randomized patients with platinum-resistant ovary cancer and high folate to mirvetuximab, we now call it Elahere, or whatever chemo you wanted to use. And the endpoint was what's called progression-free survival. How much time did each of these drugs hold the tumor stable or make it respond but not grow? How long till progression? That's what progression-free survival means. So this is the confirmatory study, and it was positive. So the primary endpoint was progression-free survival. This is the progression-free survival curve for the chemo arm. This is the progression-free survival curve for the Elahere arm. And so these are very different with what we call a hazard ratio 0.65. So to put that in words, if you have folate receptor alpha high ovarian cancer that's platinum resistant, and you get Elahere, this drug reduces the risk of progression by 35% over chemotherapy. So that is a clinically and statistically important improvement. Is it curative? No. Do we need to do better? Yes. But is this better than standard chemo? Yes, it is. Also, the thing to note is I have a little red arrow here. This is showing you the control arm. This is the unfortunate truth. And I hate to be negative, but this is the unfortunate truth of a lot of our studies that are negative, but also our standard therapies, is that by that first CT scan, which happens right where that red line is, about 50% of our patients on standard therapies have progressed. So these tumors are quite aggressive when they get platinum resistant. And we just have to do better. This is the high unmet need. 50% are off at that first CT scan. Whereas on Elahere, we see an early and maintained separation. So it's not just the progression-free survival that was better, it was also the response rate. So when you hear us talk about response rates, that's a clinical trial term. And that means when we look at the CT scan, when you come on a study, we count up the tumors and we measure them and we say, this is the sum, 10. A response means that has shrunk by 30% or more. That's a response. And so that you can see in these dotted lines right here. So all of these patients are those who responded by clinical trial definition to Elahere. That's 42% on this study. But 80% of the participants had some tumor reduction. And as a clinician who takes care of people, that's what I care about. Like, this is good. This is a win. If your tumor shrank 20% and didn't grow for 8 months, good. I don't care if it's not 35%. Of course, I'd prefer it was 35%, but these patients still benefited. And so that's what you lose when we talk about response rate. And so the counter to that, or not the counter, but the associated with that is this curve for investigator's choice chemo. You know, I showed you those terrible numbers, response rates of 13%, 6%. That sounds terrible. And it's true. Here it is. That's the response rate. But those drugs are used for a reason. They do have some efficacy. So I don't want you walking away demoralized about those as options, because these patients all had tumor reduction too. It just wasn't a response. So 55% of patients have some benefit from standard chemo. It's just better with Elahere. But it's not that the standard chemo doesn't work at all. So I don't want you to walk away with that message. Most importantly, we showed an improvement in overall survival, which has never been done in platinum-resistant ovarian cancer ever in our history. So this is the overall survival curve for chemo, and this is the overall survival curve for Elahere. And so we reduced the risk of death during the time point that we were following patients by 33%. Do we need to do better? Yes, we do. But this is the first advance in overall survival ever. And so we're very excited about this in folate receptor alpha high tumors. Now, what does that mean if you're not high, folate receptor alpha high? Well, look at the NCCN listing, and it's mirvetuximab or Elahere plus bevacizumab in folate receptor alpha expressing tumors. Where does that come from? Well, that comes from a study called Forward 2, which I'm not going to spend too much time on, but it was about 100 patients who got the combination. And they had folate receptor alpha low expressing, medium expressing, and high expressing. And of course, the highest benefited the most, and you can see them over here in yellow, but the mediums did pretty darn well. And actually, the lows did pretty darn well. And so the NCCN, it's a listing 2B. And so not all insurances are covering this. I have to emphasize it, but most are. And so this is what I have used for my patients who are folate receptor alpha medium, where I want to use mirvetuximab. And it works quite well. And we have data for that. This is the waterfall plot. We call these waterfall plots. Go chasing waterfalls. We want waterfalls to look like waterfalls. This means it works in most patients. And this is why a lot of us will try to get our patients mirvetuximab bevacizumab if they're folate receptor alpha medium, for example. And so that's on the NCCN listing. The other thing that's on the NCCN listing is all this carboplatin combination. That's new. Why? These are platinum-resistant tumors. Why would you use carbo? Well, remember I told you there's sort of different categories of carboplatin, of platinum resistance. There's those tumors that progress during platinum. There, I'm not going to use platinum again. But a tumor that responded to platinum and then I stopped it, and then 4 months later, or 5 months later, it grows back, I'm going to reuse platinum, even though they're platinum resistant, because it works. And so we can see these are a lot of studies and it's a busy slide. But going back to platinum in a platinum-resistant tumor actually makes a lot of sense in that scenario and should be remembered. And it's now on the NCCN, which I think is important at reminding our providers about that. So that's standard of care. Let me bip through the clinical trials portion of this. So I'm going to talk about biomarkers and sort of what's coming. Biomarkers are really important. I'm just putting this here. I know I'm talking about platinum resistant, but everyone should be offered testing for BRCA, 100%. So if you have not been offered testing and you have epithelial ovarian cancer, you need to be tested, unless you don't want it, and that's fine. But if you do want it, you should be tested, 100%. We are not at 100% in the US, and it makes me angry. And so if you have not been tested, please get tested or ask for it. It's on every guideline. We all agree that all of you should be offered testing. And if you want it, you should be able to get it. It is paid for. But there's more than that now. So these are all the biomarkers we're looking for now, especially in the platinum resistant, but even moving up into frontline into the platinum sensitive recurrent setting. I talked about folate receptor alpha. We're looking for biomarkers of immunotherapy. We're also looking for all of these proteins on the surfaces. I talked about folate receptor alpha, but we're looking for all these now. HER2, cadherin 6, that's what CDH6 is. There's even more. So the biomarker picture is getting much more complex, and we're having to retest patients that got tested maybe 2 years ago as new things come out. So we make sure we can get patients the right drug at the right time. This is sort of just a busy slide to show you all of the clinical trials that were active as of a few weeks ago. This has changed. I'm going to show you in the platinum-resistant space. There's antibody-drug conjugates, immunotherapy, drugs that target how tumors make blood vessels for themselves, and then other sorts of targeting agents. So much effort to try and do better. But this is a hard place to do better. These are no longer here. These were all negative. So it's a hard place to show improvement. But antibody-drug conjugates are really our kind of leading opportunity here, these targeted chemotherapy agents. So I just want to show you what's coming. So this is sort of just rapid, just so you're excited about what's coming in antibody-drug conjugates. I've told you about mirvetuximab, which is targeting folate receptor alpha. There's at least 4 additional ones coming behind it. This is luveltamab. We call it STRO-002. It targets folate receptor alpha. It has a different chemo on its tail. So it's not the same drug. This is its phase 1 data. You look at the waterfall. Waterfalls should go down. That's good. So we saw a lot of benefit. It does have some different toxicities, and the company's working through those, but this is moving into a phase 3 trial. Farletuzumab acetaretin, it's hard to say, targets folate, has a different chemo on its tail. So this is a different drug. The phase 1 data, all in ovary, looks pretty good. That's a nice waterfall plot. It has different toxicity than the other 2. These 2 are currently in phase 1 studies. This is my world. These are the 2 I have open right now. Completely different drugs, different targeting of folate and different chemos. Why do we need this many things targeting folate? Well, just like breast cancer has like 4 drugs targeting HER2. And they use one. When it stops working, they go to the next one. And then they go to the next one, and it keeps working. So if we show that you don't lose the folate receptor when you progress on mirvetuximab, then I'm going to use one of these drugs because it has a different chemo on its tail. So if I can just deliver a different chemo to a now resistant tumor, I may be able to keep targeting folate. So just because you've had one folate drug doesn't mean you may not get another. Time will tell. Cadherin-6 is another emerging target. We don't even have to test your tumor. It's so prominent. So we may not even need a biomarker here. Time will tell. We're still working on this. Different chemo on its tail. This is the phase 1 data from last year. I'm going to present the updated data in a few weeks at ESMO, so I can't show it to you yet, although I'll tell you it looks better. And it's been very well tolerated. This is moving into a phase 2-3 study. So watch for this. This is coming in ovarian cancer as well. We're really excited about trastuzumab deruxtecan. This is in HER2, all over the breast cancer literature, as just a game changer, because the biomarker went from being HER2 high for Herceptin, now any HER2, 1+ and up, benefits from this medication in breast. Does it work in other cancers? Destiny Pan-tumor was presented this year at ASCO. These are all the tumors that were included if they had any HER2 expression, 2+ or higher. And we can talk about that in questions. But here's the ovarian data, and I can show you tons of it, but it works. That looks pretty good. 63% response rate if you're HER2 3+, 37% if you're 2+. I mean, that's better than anything we're using chemo-wise right now in the platinum-resistant space. And you can find HER2 positivity in about 30% of high-grade serous ovarian cancer, 1+ and up. So we have work to do here, and we're designing the trials to bring these forward. So a lot of antibody-drug conjugate work coming. The other place, and then I'll end because I think I only have 2 minutes, is immunotherapy. This is from Catherine Fu, who's my friend and is brilliant. Immunotherapy is hard because ovarian cancer, for whatever reason, creates this shell around it that is so hostile to our immune system, like doing its job and killing the cancer, that we can't get the immune cells in there to kill the cancer. So typical things like you see on TV, pembrolizumab, which is Keytruda, Opdivo, which is nivolumab, by themselves just don't work. This is why. And I'm not needing to explain this, but it's so complicated. There's so many mechanisms of resistance in ovary cancer that just a standard immune checkpoint inhibitor is not going to cut it. And we've seen that time and time again. It's not working. Combinations may work though. And this data is from our friend, Dr. Zeros, and it's oral Cytoxan, which is an oral chemo, low dose with Avastin with Keytruda. I'm going to use commercial names. And this is the waterfall plot. So this isn't FDA approved. It'll never be FDA approved regimen, but it works really pretty well. And I use this all the time. So I'm showing it to you. Like sometimes we figure things out and we use them. Combinations may be key with bevacizumab. There are 3 studies that are in the phase 3, not with Cytoxan, but with more modern chemotherapies that we'll see. Does this triplet strategy work? And these are the 3. And these will all start reading out. You'll see this 3rd one read out in November. That'll be the first one of these chemo, immune checkpoint inhibitor, bevacizumab triplet versus chemo, bevacizumab alone studies to read out and whether or not this moves into standard of care. So exciting. New chemotherapy, new immunotherapy options are needed though. It can't just be the Keytrudas. We need novel things that are gonna get your immune system programmed to fight something that is tumor specific and expand those cells massively to really get that immune system in your body. And we're either gonna do that by giving you something that does that. This is one example. This is nemvaleukin, which is a modulated interleukin-2 that basically works to try and upregulate the fighter jet T cells and what are called natural killer cells in concert with pembrolizumab or Keytruda. This is in a phase 3 study right now. So it's a novel way to come at the immune system. This is, I know many of you have heard CAR T. This isn't CAR T, but it's like that. It's adoptive cellular therapy. This is where we test your blood. You can see it over here in this figure. Here's your cancer cell. It presents a protein on its surface with this specific thing called HLA. So we have to test your blood to see if you have HLA-0201. That's about half of Caucasians. Varies by ethnicity. Has nothing to do with your cancer. It's just what you're born with. So we target HLA-201. And then this particular product targets a protein called MAGE-A4. So we test your tumor to MAGE-A4. If you have those things, then the company makes many billion cells of engineered T cells that target MAGE-A4 out of your T cells. And we give it back to you in a big bag one time to try and create this massive influx of immune cells that are targeting your cancer, a protein that's on your cancer, to wipe it out. And the current study that's ongoing is SURPASS-3, which is the cells alone versus the cells plus an immune checkpoint inhibitor to try and maintain the benefit for longer. But you treat and watch. That's it. It's one time. Response rates in phase 1 were 42%. Small numbers and highly selected. But this is open and accruing. And so you could potentially be eligible for that. And the last one is really looking at intraperitoneal immune therapy. We thought we were done with intraperitoneal stuff, but maybe not. This is a vaccine that really tries to trigger immune triggered cell death intraperitoneal. So it's really for intraperitoneal tumors. This is the phase 1 data. Look at the waterfall. It's all going down. So these were selected patients. Why do I say that? I'm coming back to the platinum conversation we had. These are patients that likely got platinum, responded, and then grew back really fast. Because it's platinum plus this intraperitoneal immune conjugate. That's where you saw those responses. So this is the phase 3 study. Platinum alone in platinum-resistant tumors versus platinum plus this intraperitoneal immune conjugate. And this is open. This is called Ovavec. And this is open and accruing across the US as well. Novel immune therapies. Keytruda is not going to cut it. I love Keytruda. Not going to cut it. We've got to do things in a different way. So short and sweet, although my talk wasn't short and sweet. Platinum-resistant high unmet need. We've got to do better. Trials are the key. And really a whole nother talk is the best way to treat this is to prevent it. So if we can prevent cancers in the first place, and we're working on that. B, if we can prevent recurrences in more and more women so you don't fall into this category, that's going to be the best way to treat this. In the meantime, we'll keep coming up with things that hopefully work better than Doxil. And with that, I'll turn it over to my colleague, Dr. Fader.